of the end-binding protein with just the distribution of the DNA. And we do this in a very similar way, but we use an antibody tagged with a different fluorescence group so that now it glows green instead of glowing red. We see that the DNA also fills up most of the nucleus. And when we superimpose these two images, yellow tells us everywhere where the telomeres and the DNA coexist. And you can see the nucleus has co-localized chromosome ends and DNA, except for a number of holes which have neither DNA nor telomere protein present. I'll get back to those holes later on. If we do the same experiment with the other subunit of the end-binding protein, from what I've told you about it, the beta subunit is right there, hanging on to the same complex as the alpha subunit. So if we've done the experiment right, it better be in the same location. And sure enough, beta fills up the nucleus along with the DNA and the superposition of the two. This is a different nucleus, so it's a little different shape. I call this one the eggplant. But in any case, you can see that it fills up the entire nucleus, except again for a number of holes which contain neither DNA nor telomere protein. Now let's switch over to asking the question, where is the telomerase located within the cells? The telomerase is the enzyme that's going to copy the chromosome ends. So we assume that it, of course, would be located in the same place. Is that the case? So what's the technique that can be used? Again, there are many different RNAs within a cell nucleus. There are messenger RNAs, transfer RNAs, ribosomal RNAs, and a very small fraction of the RNA is this telomerase RNA. We want to find the location of this RNA and not be bothered with all of the others. We can take advantage of the specificity of Watson-Crick base pairing, a theme that's come up probably 20 times already in these lectures, A recognizing T or A recognizing U, and we can make a probe of a sequence that will be complementary to and thereby bind to the telomerase RNA and not to the other RNAs in the cell. That probe molecule can be chemically derivatized with a small molecule called biotin. The reason for doing that is that there's a protein that's quite easily obtained called avidin that has a very high affinity and binds biotin very specifically. And avidin can be chemically derivatized with a fluorescence group. So it's the details are different, but it's a very similar strategy to how do we tag a specific protein in a cell. If we want to tag a specific RNA, we use a biotinylated oligonucleotide, a stretch of nucleic acid sequence, to pair with the RNA of interest and in turn recognize where that is in the cell by a fluorescence-tagged protein. When that experiment was done on the Oxytricha nuclei, we see a pattern that is strikingly different from what I showed you before. You can see that there is fluorescence throughout the nucleus. By the way, for those of you watching from Texas, this particular stain is called Texas Red, which I think is a great name. You can see that there's Texas red staining indicating where the telomerase RNA is located, to some extent throughout the nucleus, but particularly in a number of globular focal points. And then there's this band. It looks sort of like a belt around the nucleus. That turns out to be the site of active replication. So the replication in these nuclei starts at one end of the nucleus, moves to the other, and active replication is taking place inside of the replication band. And you can see there's quite a bright staining for telomerase RNA at that point. So now let's look at where the telomerase is located relative to the telomeres. And as I said before, our expectation would be that they would be in the same place. Instead, they're almost completely segregated from each other. Here's where the telomerase RNA is located. Here's where the telomeres are located, as judged by the alpha antibody or the anti-beta antibody. And when you superimpose the two images, you see that the telomerase is filling up those holes where there isn't chromosomal material. Here's another example. And only in the case of a replicating nucleus, where we have a replication band, do we see telomerase in the replication band, we see DNA and telomeres in the replication band, and we see a nice yellow stripe at one edge of the replication band. So perhaps we've learned something, or at least we have enough evidence to come up with a reasonable hypothesis that maybe telomerase works in a concerted way, coordinated with replication of the middle parts of the chromosome. And that's why the two sorts of molecules are localized in the same place at the same time. Well, then how about all the telomerase that's localized in these holes where there is not chromosomal material? Well, maybe that is a site of assembly of the telomerase, where the RNA is assembled with the protein subunits to form the active enzyme, or perhaps it's a site of storage or transport or maturation of this telomerase ribonucleoprotein enzyme. You may think it's strange that the nucleus devotes so much of its space to just storing telomerase, but and we thought it was very strange, and therefore we said, well, I'll bet there are other things being sequestered in those spherical areas as well. So it turned out that other small RNA protein complexes, such as the SNRNPs, the SNRPs, which some of you have asked me about at the coffee break, which are involved in messenger RNA splicing for those introns that aren't self-splicing. They have these other RNA protein complexes that are involved in their splicing. Those are also localized in these same sort of spherical bodies as the telomerase. So these are not, if these are storage or maturation depots within the nucleus, they're not working only with telomerase, but with a number of small particles that have to have RNA and protein organized in a very cooperative way. So in summary, we find that this protein that caps off the end of the chromosome co-localizes with the DNA, that much to our surprise initially, most of the telomerase doesn't co-localize with the telomeres, but instead is found in these spheres that also contain these SNRPs, and they may be assembly or storage centers, although that's a speculation that remains to be tested. Finally, we find that telomerase does co-localize with the telomeres at the site of active replication. And to the extent that two things being together implicates their acting in a coordinated way, which again has to be tested, we can at this point only say that we can suggest, rather than that we've really proven, that the replication of the chromosome end is perhaps coordinated with the replication of the middle parts of the chromosome. So I've taken you in this last hour through a sequence of events that have allowed us to look a little bit more about how a portion of the cell is organized, how the nucleus is organized with its chromatin, which is the DNA, chromosomal DNA, rather dispersed through the nucleus, but there is some substructure in the nucleus, and that some of that substructure is involved in handling the enzyme which is responsible for replication of the end of the chromosome. I've also told you something about DNA-protein interactions and how those DNA-protein interactions can protect the chromosome ends from undergoing unwanted degradation or recombination reactions and thereby stabilize these linear pieces of DNA. We imagine, but as yet have no direct evidence, that the very ends of human chromosomes are perhaps also stabilized by a specific protein, although that protein has not yet been found. Obviously, that's a much tougher problem because there are many fewer chromosomes and many